Thank you, Sally. It's now my pleasure to introduce today's plenary session. Changing landscapes of plant pathology highlights two factors that will have a profound impact on the science and practice of plant pathology: new technologies and the next generation of plant pathologists who will apply them. This session will feature three presentations on some technologies that will have a substantial impact on our science going forward. Our first presentation by Greg Heck, Science Strategy Operations Manager at Monsanto, will present components of successful utilization, including efficacy, robustness, specificity, and safety of double-stranded RNA as an active agent. A historical perspective, advanced examples, and prospects. Please join me in welcoming Greg Heck.
Well, thanks, Tim, and thanks to the organizers for choosing this subject matter, which has been near and dear to me for my research for quite a number of years now, being at Monsanto and working both in our insect control and basic tool development around these technologies. I'm only given 25 minutes here, but I could probably talk 8 hours, which is probably exactly why I'm not invited to parties. But we'll see what we can do, and I'll try to stay behind the podium since I'm kind of tethered here. Although they told me that there are wheels, and I could do a Melissa McCarthy kind of thing, doing Sean Spicer, but I'll refrain from that as well. But let's move forward. And my title here says RNA technology, and that was an intentional and purposeful sort of use of the term versus RNAi, which is what I'm going to focus on. So RNAi is but one constellation of technologies and, if you will, interactions of sequence-based recognition amongst RNA molecules that can affect gene expression, both in suppressive modalities as well as augmenting expression. So we're going to focus today on RNAi, and we can go a little further into that and sort of ground you on what it is that RNAi is. It's often used as a term, but there's a lot of understanding behind it. And that understanding started probably in the early 90s with Jorgensen's work and others that were looking at overexpression of pigment genes in things like petunias. There they were trying to intentionally overexpress shell cone synthase, which is a primary determinant of that pathway. And what they got back out was some darker petunias, greater pigment production, but they also got a variety of petunias coming back from transformation that were various patterned petunias. That star-shaped pattern that I showed you in the previous slide, that's a natural example that is present because of RNAi. But they were able to do it transgenically. They got all the way to white flowers even in some cases. And when you examine the RNA transcript for shell cone synthase, it was absent or extremely underrepresented in those white sectors. So they were doing something. It was a phenomenology. It was called things like co-suppression. The underlying mechanism was not known. But subsequently, Fire and Mello, who were working with the nematode C. elegans, found out that RNAi was present and that double-stranded RNA was a key determinant in this process. It was the common currency, if you will, for it. Cells in higher organisms recognize that double-stranded RNA has a special property and utilize that to then target gene suppression. So there are enzymes that will break down and reduce double-stranded RNA into smaller usable units, typically around 21 to 24 base pairs in length. And those can be programmed into cellular machinery that then use that information to scan RNAs and target them. They may just sort of sit on those RNAs and stop them from being translated, or in many cases, in terms of plants, actually lead to cleavage of those molecules, thereby reducing gene expression. Naturally, this actually has a lot of purposes in the plant. That is one One modality would be trimming gene expression, controlling the fine points. So promoters and transcription factors and things like that definitely have a big sway in determining gene expression in plants. But microRNAs and the associated small RNAs that plants naturally produce to regulate their genomes modify that further and provide the fine-tuning there. They're also used in things like epigenetics or genome management even, controlling transposable element activity are all within scope here. And then there's also the viral invaders. So plants can mount defensive actions against viral invaders, targeting the sequences that are present from these incoming foreign nucleic acids. So there's this natural system. It's in place. It's waiting to be programmed. The question really is, what do you do with it? And as technologists, we can look to that and say, I can provide, assuming there's a suitable route for providing it, the information that would target a gene of interest. And so that can be a number of different things. We have a variety of tools available to us at this state of the game. We understand how they function fairly well. You might have to do a little bit of work to get them in your particular system, but they're available. You can produce a single small RNA with something like a microRNA backbone, which is, again, one of the natural things that plants would utilize in their own genome. But we can substitute out sequences of our interest. So if you wanted to target a particular enzyme or pathway that might be involved in a plant defense response, you could modify that using a microRNA, for instance. If you wanted something more robust, you might have to choose something like a hairpin construct, which would provide a constellation of small RNAs that would buffer maybe sequence variation that might be there in a viral population. And so we can do that these days, and probably the most exciting examples that are current use the hairpin structure. So the co-suppression type of methodologies really relied on overexpression and the survey mechanisms in the cell to recognize those as aberrant RNAs and try to suppress them. Creating an intentional structure that folds back immediately upon itself is extremely efficient in generating those small RNAs. And that's probably where we go in most cases when we have to choose that pathway today. But what you can get when you get there is a variety of outcomes. You can get deep suppression. So you can actually essentially almost eliminate a particular transcript from the genome. But in other cases, you may want a shade of gray. So this particular example, we modified soy oil composition by turning down a couple of desaturase enzymes that are operational in the seed and changed soybean commodity canola oil to, in the Visitive Gold product, something that looks much more like canola. So we shifted things into the 18-1 unsaturated fatty acids, the oleic acids, which then gives a much better utility to the soybean oil. And that was done by partial suppression. If you deeply suppress these particular enzymes, the seeds don't germinate. So it's a really bad day for your crop. But you have to sort of compromise. And so we found that by scanning through, choosing just the right segment, in this case an intron, for instance, might be the way to get just enough suppression, maybe about 80–90% suppression, but not complete suppression. So in short, we have a system, we have a variety of tools to get the RNAs produced in planta if we're targeting something within the plant cell. We can program it with a bit of experimentation, the right sequence, and get a very specific outcome there. But you can do more with it. By actually actively programming that against a plant pathogen like a virus, for instance, we can affect very deep and protective levels of suppression. And so the really long-standing and shining examples here are in virus and squash, or papaya and squash. And they've been on the market for over 20 years, and probably longer if you count the time and development that they've been in the field and have been durable in their resistance. I think it's pretty exciting that you can get that level of protection over those many years. That was probably largely done because a large amount of real estate, multiple small RNAs were being produced, which buffer the potential for mutation in the virus and lead to that long-standing suppression. But you don't have to stop there. You can choose more sophisticated means to get there. This is some internal proof of concept work that we had done with tomato, where we weren't just choosing to suppress a single virus, but we were choosing 9 viruses within a single transgenic locus. We had 2 different transgenes in this particular case, back to back, but they were producing hairpins that were targeting both DNA and RNA viruses. There's some art there, of course, because you have to choose the right segment of the virus. In some cases, it might be the rep protein. In some cases, it might be a coat protein. And we had to do experimentation to find out which was most effective for the given virus, and put in enough sequence. So here we were using approximately 300 base pairs of target per virus. So we had done deep sequencing in native field populations to understand how much natural genetic variation. So when you're targeting something with a gene sequence, you want to make sure that sequence is sufficiently accommodated for any natural variation that might be there. So going too small tends to lead to a more precarious outcome in terms of long-term durability. But you can get there, and you can get very deep levels of suppression. In this case, the yellow leaf curl virus, we can get deep suppression, and if you inoculate one leaf, you can take a distal leaf and do PCR, and you cannot detect the virus. Which, if you look at some of the natural resistances that are available, you can see a low level of transmission through the plant for that virus. So we can get extreme suppression. And if you imagine implementing this on a broader scale in a regional context, you might be able to suppress that virus to a very high degree regionally. So, if there's one thing we know how to do with this technology, I would say it's viruses. And we definitely have opportunity to do this in a lot of crops that would benefit. But you can go even further beyond this. You can look outside of the plant. So the virus, the oil modification, maybe targeting a pathway involved in defense would all be inside the plant. But what Fire and Mello did, in addition to discovering the core elements of the process, they found that environmentally, or eRNAi, could be possible, where C. elegans, which was their model system, could consume a bacterial host that was producing double-strand RNA, and through uptake in its gut, take that double-strand RNA, distributed it between cells, and affect gene suppression. In this case, they determined that there were a variety of SID genes that are actually going there. You have to remember, RNA is a large, double-stranded, charged molecule. It doesn't transit a membrane just by accident, but needs to be that in a facilitated way. And so C. elegans had that system inherently there, and you could play into it and target it. And so parasitic nematodes have definitely been of interest in the literature for folks looking to say, can we extend this? And some species are probably going to be tractable and some not. And maybe if I have one message is you have to know your organism really well, because even very closely related Caenorhabditis species don't necessarily have the uptake mechanism. So it's going to be dependent upon the organism in part and whether your delivery mechanism can get there. But once you do that, you can look at other invertebrates, like the vectors, perhaps. In this case, and I'm going to steal my thunder and tell you it actually really works in Planta, we had engineered against the beetle larva, corn rootworm, a double-strand RNA that suppressed a key gene involved in a core pathway in the insect, and we're able to suppress it enough that the insect stopped feeding and died. And so we thought there's a new way here to go forward, and we're actually in a very advanced state of that project and have most of the regulatory approvals done. And it's been a real nice learning experience for the technology, and I would say in many cases, if you look to other organisms that are responding to environmental RNA, you're going to see very similar things going on here as well. And so where are we with that? You can engineer the plant, or you can spray it on. And the engineering path is going to be the path for something like corn rootworm, where you have to get it into a below ground state where the larva feeds. And the insects that are responsive in this space, I would say, run a gamut. So corn rootworm is really sensitive. So here we're talking parts per billion response to RNA. You can work with that in a plant. If you go to something like a whitefly, the literature reports will tell you have to jump up probably an order of magnitude or more in terms of the amount of RNA you have to supply to that insect to get an impact, all the way to something like a lepidopteran, a moth larva caterpillar, where I would say it's recalcitrant. You can put 1,000 times or more the amount of double-stranded RNA equivalent that it takes to control corn rootworm, and there's no impact. This is a spectrum. The beetles are probably the most impactful in terms of impacted in terms of the orders of insects that have been tried. So if you choose some of these other targets, you might have to figure out creative ways of getting the RNA in concentration to the insect in question. That might work in some cases where there are baits for the insect to consume. But rootworm feeds directly on the plant. Unlike the lepidopterans, which have very aggressive nucleolytic environments in their gut, and from all of our determinations, we don't see the uptake mechanism being in place. Corn rootworm does have it. It has a more friendly gut. The RNA that's produced in the plant has time to get there, and then once it's there, it's taken up and distributed in the body of the insect with a dependency of size. So you have to be at least 60 base pairs or larger in size to get it into the corn rootworm. And so that's a key feature. Not only do we need to know our target sequences, the population dynamics, we also have a constraint around size. And I would venture to say that most of the responsive invertebrates are going to be in this kind of bucket. And what's really cool, and again, this is why I'm not invited to parties, but we went back and reanalyzed data. And I totally geeked out when Sergey Ivashuta came and said, hey, look at this. We had dissected out corn rootworm guts to remove all of the plant material and sequenced the body of the insects to look at the response from an RNAi perspective at their small RNA populations. And we sequenced corn rootworm as well, which is what they were feeding on. And we asked the question, well, what happens if you actually map back to the corn genome in both cases? And what's pretty cool is you can image the double-stranded RNA content. So plants naturally have a lot of double-stranded RNAs that they're producing, again, for controlling transposons and so forth. Here's a small segment of chromosome 1. The height of the line indicates the amount of small RNAs being generated. But there's corn. But you can image corn by sequencing rootworm. So rootworm is non-specifically taking in all of the double-stranded RNAs and processing them as 21 mers versus corn favors 24 mers in this case, and processing them. So it's seeing literally no sequence discrimination. It's taking those in and utilizing them. And so if that's the case, that means we could supply any given target sequence we think is going to actually control the rootworm. So there's a system, it's programmable, it's not sequence discriminatory, so we can provide that sequence that does control. And the bottom line is you can get there, and we can control corn rootworm in a field scenario with high levels of pressure and actually affect root control. And we combine this with some BTs, so we have buffering for resistance development, but it's been a pretty strong and wonderful outcome. But you don't have to stop there. You can spray this on. In some cases, there'll be targets that are able to manage through and encounter that RNA in a productive way and take it up. There's hurdles, so it's not trivial. This stuff tends to go away. You got to be able to produce, if you're thinking broadacre, metric tons of RNA, and so that's not trivial either, but I think we've solved a lot of those kinds of questions. And then it's really coming down to the system at hand. And one example of that is mites. So we looked at honeybee health, and mites are broadly responsive to double-stranded RNA in their diet. And we thought varroa mite, which is a key determinant in a lot of the colony collapse disorders, it's a vector for many viruses in honeybee colonies. We looked at it relative to apivir, which is actually a commercial miticide, and found that double-stranded RNA in some of our early trials could actually produce a lot of protection against the phoretic mites. I'm sorry, the brood mites, not the phoretic mites. So they have two different points in their life cycle. And what we found, and again, knowing your organism, was that honeybees were distributing it not through their own bodies, but it was taking in a sugar syrup that we were giving them and depositing it in brood cells for the larvae where they're attacked by mites, was the key way that the mite was encountering it, in the brood food and not through the honeybee itself. So that sets some sights on how we need to try to figure out to stem the longevity of this so it can actually be productive in a long-term use scenario. But you don't stop there. So we know it's a good technology. We know that it has a lot of potential applications, but you want to make sure, particularly since we started looking at insecticidal types of control with RNA, it raised a lot of questions about how this is playing out in the environment, the durability, and so forth. And that became a big question because when you went into the literature, you found often, everybody knew that RNA was fairly fragile as a molecule, but nobody really ever published how fast it goes away in soil. So we had to look at those things amongst others with specificity as well. So I think Jack Bobo said it's hard to convince people with facts. So you're a science crowd, so I think I'm going to help myself along a little bit by hoping you're going to follow this. But we're not putting much into the environment, which is like a starter. And you can sort of argue with my calculations a little bit. And even if I'm wrong somewhat in terms of the root mass, I didn't have a real fine measure on that. But we knew a lot about the above ground masses, and we certainly knew the different plant part abundances of double-stranded RNA that we were engineering into the corn rootworm product. And the long and short of it is there's not much out there. There's about 9 kilos or so in a field of corn plants, but 35 milligrams of that is our transgenic RNA. So think of any biologically or chemically active pest control substance that's working at 35 milligrams or so per acre. It's pretty amazing. But it, again, plays into the exquisite sensitivity that corn rootworm has for perceiving the presence of RNA. You have to know where everything is. So we've developed the methodology to quantify that, so I could actually do that calculation. We needed to determine where and when, how much of it is present, how long it persists. The good news is what you would think is that its persistence in biologically active soils, water columns, so forth, is pretty short. And the bioactivity goes away with the loss of RNA. And so we're looking at that. We also looked at, well, okay, you're not putting much into the system, but maybe it's overwhelming. And so again, we're sequencing corn rootworm that had been fed corn. And what we found was that a very significant portion of its double-stranded RNA, the small RNAs that it was present in the insect itself, were actually coming from corn. So I thought this was pretty astounding that more than 10% of the 21 base pair population of RNA was actually not from rootworm, but from corn. And somehow it's learned to manage that. We weren't able to see any impacts itself from the corn plant, non-transgenic or transgenic. But when we looked at a transgenic neutral sequence put into that system, we found that it still made up a very small fraction. In this case, about less than 2% of the total small RNAs were actually coming from our transgene. Most of it was still from the insect. Most of it was still functional. But if that was a targeted sequence, one that should have targeted a particular message for shutdown, it would have had a profound effect because those shots on goal really matter. So the stoichiometry, how many exact sequence matches you have and how abundant they are, makes a difference. You can look at specificity. So we did reciprocal feeding studies looking at corn rootworm versus closely related species. And we can't assay all of these things, but we could look at their sequences and feed them back to corn rootworm and say, how could a related species that might only have 3 base pair or 321 matches out of the total sequence that we are looking at, could that affect a regulation in that gene? In that other organism. And we found that you really needed an exact match. So there's a lot of data and literature out there saying that less than perfect matches could have consequences. But what we found, feeding high levels of double-stranded RNA with many different variations of match, it was really only the exact 21 mers that really mattered. We could do the reciprocal feeding studies, taking Colorado potato beetle, feeding back other sequences. The same story came out. You needed at least a few 21 base pair exact matches to really affect gene expression. Plus, you also have to have that organism directly feeding on the corn plant itself. And so we looked at other organisms, looking at a wide variety of things that might be found, or surrogates for things that would be found in a field, parasitoids, and so forth. All of these things, feeding hundreds or thousands of times more double-strand RNA, we had really good profiles. We looked at multiple life stages. In the case of honeybees, we looked at pupation, emergence, adult weights, longevity. Really weren't able to find anything when the sequence was well chosen. And so there's again, a lot you have to go there and look through, but it's tractable. From the mammalian consumer standpoint, there was a lot of concern in the literature that mammalian consumers might take up small RNAs from diet. It really hasn't been substantiated in ongoing work. And it's not for want of trying. The pharmacology and world has been trying to get the RNAi pill to work for some time. They've had success in injectables with modified double-strand RNAs, but certainly not through feeding modalities. But we didn't stop there. We actually did a full assault with some mice and some rats, feeding them their own exact sequence. So this is giving them as much small and long double-strand RNA matching as possible against one target gene. Looking at a variety of pathology endpoints, weights, and so forth, we weren't able to see any impact, which agreed with the understanding that there wasn't significant uptake in mammalian systems. So that probably brings me to the end where, I think we have a good message here. We have a fairly mature technology. It's really not, unreasonable to be able to get your particular small RNA produced in a plant. The real question is, will the biology that you're interested in support the use of that? So is there a route of delivery? Will that pest or pathogen actually encounter it in a meaningful way? You don't have to do everything by transgenesis. Certainly some of this might be tractable through sprays of double-strand RNA. And that particular control methodology is probably something that's going to be really useful because of its environmental and specificity properties we have out there. So with that, I'll let it go, and we'll talk about questions later. And I'll be around for a while, so if you have particular questions about some of the things I didn't talk about, we can have some really good conversation.
Our second speaker, Erica Goss, Assistant Professor in the Plant Pathology and Emerging Pathogens Institute at the University of Florida, will describe how population genetics and genomics can help identify pathways of movement, genetic shifts in local pathogen populations, and ultimately changes in disease observed by pathologists in the field. Please join me in welcoming Erica Goss.
Thank you, Tim, for the invitation to speak today. I just want to take this opportunity to say how grateful I am to be part of the APS community and all the amazing things you're doing. So, as plant pathologists, we often work on problems that are local to us, that are in our own backyard, in our state, or problems that are in someone else's local area. For example, if we're working internationally. And plant disease is so influenced by the environment that it really is inherently local. But disease is also influenced by global processes, for example, global climate change, and also by long-distance movement of plant pathogens, like I'll be talking about today. So To understand our changing landscape of plant pathogens, we need to look at the local scale, and the global scale. So that's what I'll be talking about today. So plant pathogens have geographic distributions, most of them determined by the distributions of their hosts, by the abiotic and biotic environment, by their history of dispersal and migration, and by local evolution, among other factors. We know that plant pathogens have geographic distributions when they go somewhere new, when they colonize a new region or a new host and cause devastating disease. But disease severity can change due to pathogen migration, due to movement of races, fungicide resistance genotypes, or any other new genotypes that might cause a change in that might be more aggressive. So for example, UG99 is an example of a spread of a new race. And for Phytophthora infestans, we've repeatedly seen replacement of resident genotypes by new genotypes. So if we imagine that there's variation in disease across a landscape, we have a sort of mosaic, a geographic mosaic of disease. So in region 1 and region 3, in this simple example, we have the plant disease triangle fulfilled, so to speak, and we have active disease. Whereas in region 2 and region 4, those are cold spots. There's no disease there at this time. So depending if you're in region 1 or region 2, or 3 or 2 or 4, your priorities for research, extension, and regulation of this disease may be different. So when a pathogen moves from one region to another, this can shift this mosaic of disease, because now you have a pathogen that's a virulent race, or is more aggressive, or more adapted to the climate in that region too. So this can dramatically change local priorities. So if we see a dramatic change in the field, why do we need population genetics to study this obvious change? Well, often we don't know that there has been pathogen migration. And we're skeptical scientists. We don't like to jump to conclusions. So we often go through the process of thinking about many different reasons why we could observe this change in the field. So the first step is often to just identify that there has been a change in the pathogen distribution or pathogen population. And this is a figure from David Cook's work on Phytophthora infestans in the United Kingdom, where he showed a shift in the genotypes over time to this aggressive blue genotype called 13A2. So by 2008, this was the dominant genotype in the UK. Fast forward to 2016, and it is now this pink genotype that is the dominant genotype, whereas the aggressive blue genotype persists in other parts of Europe and has spread to other areas, including Asia and Africa. So once we identify that there's been a change, we may want to identify the pathway of movement that brought that new pathogen or genotype to that location. So this is work I did with Nick Grunwald on the sudden oak death pathogen Phytophthora murmurum, and we used population genetics to infer migration pathways of this pathogen. And for the lineage NA1 shown here in yellow, we were able to confirm our population genetic results with APHIS trace forward records to show that our genetic results were actually valid and could reconstruct migration pathways. So a more recent example comes from the emergence of wheat blast in Bangladesh, where they used a population genomic approach to show that the genotypes of wheat blast in Bangladesh are as similar to genotypes found in Brazil as genotypes found in Brazil are to each other. So basically, this was could have been part of the Brazilian population, suggesting a pathway of migration to Bangladesh. So we might be interested in some pathways of movement more than others. For example, we may be more concerned about migration from populations with high genetic diversity than from populations that have the same genotypes that we already have locally. So I was interested in looking at populations of Phytophthora infestans in central Mexico, which we know is a center of diversity of this pathogen, and we suspect that the genotypes we find in the US have come from Mexico. So we did population genetic analysis on isolates collected across central Mexico by my collaborator, Silvia Fernandez Pavia. And our results are shown in this plot on the right, which is like a principal components analysis, but we're maximizing the difference among the states where we collected. And you can see that it mirrors the geography of Mexico. So we did find genetic differentiation from populations in different regions. The blue circles on the left are from the western state of Michoacan, whereas these green circles on the right are from the eastern part of central Mexico. So then we projected the US genotypes on top of this plot. And what we found is that some lineages have genotypes that are very similar to what we see in Michoacan from western Mexico. Others look like they came from populations we didn't sample. But the US 23 genotypes actually grouped with the eastern genotypes. So this suggests that there have been introductions from multiple regions of Mexico to the US and suggests that there may have been more than one migration pathway from Mexico to the US. So in addition to sort of reconstructing historical movement and demography of pathogen populations, we can actually use this together with studying selection on genes important for disease management. So an example of this would be fungicide resistance, and this is a very simple example, simplified example here. So in region 1, we have fungicide resistance in our pathogen, but not in region 2. So if fungicide resistance emerges in region 2, is that due to local evolution or introduction of resistance from region 1? We do our population genetic analysis, and if we find that migration occurs from region 1 to region 2, then it's likely that this was an introduction from region 1. But if we find, and then if we can block that migration and maybe return the population to sensitive state. But if we find that the migration is occurring in the other direction, exclusively in the other direction, well, then it seems more likely that this is an instance of local evolution of fungicide resistance. So this is very simplified. In real life, it's more complicated than this. And I want to talk about some results from Mason Newark, who just received his PhD at UF from working with Matthews Perret and Nick Dufault. And he was studying fungicide resistance in the gummy stem blight pathogen on watermelon. So there's fungicide resistance in Florida. And the question he wanted to ask is, could this be coming in on seed rather than a locally developing problem? So ideally, he would sample the seed production area, but that wasn't possible. So alternatively, he had the opportunity to go to fresh market production areas in China and sample there with the idea that they are getting seed from the same source as Florida is. And so if he sees fungicide resistance in China, then chances are we could be getting it from the same place. Of course, this is not what he found. So China has different fungicide resistance profile than Florida. Their profiles in Florida and China correlate very well with the different management practices in these two different regions. But we also did population genetic analysis on these strains. And he used microsatellites developed by Maren Brewer at University of Georgia. And what we found from those is that there was no genetic differentiation among populations in Florida and China. This was really surprising to me. It really looked like they were one single population on two different continents. So here we have a pathogen that apparently is moving around a lot. Meanwhile, we have different fungicide resistance profiles in both regions, suggesting that it really is likely to be a local problem managing this fungicide resistance. So this system is a great candidate for population genomic analysis, because then we can really tease apart the effect of migration and pathogen movement among these populations from the effect of local selection and evolution. So hopefully we can continue working on this question. So now I want to move to a true population genomics study. And this study is looking at a race shift in Xanthomonas perforans, which causes bacterial spot of tomato in Florida. And this is a collaboration with Gary Villard and Jeff Jones. So just a little bit of background. Xanthomonas uvesicotoria, which is closely related species to Xanthomonas perforans, was the causal agent of bacterial spot on tomato in Florida in the 1980s. In 1991, Xanthomonas perforans came in and displaced uvesicotoria from tomato, and we suspect this is due to the bacteriocin activity perforans has against uvesicotoria, shown on the plate here as the zone of clearing of the uvesicotoria lawn around the colony of perforans in the center. But in 1999, perforans tomato race 4 was found. And by 2012, race 4 had taken over the population, completely displaced tomato race 3. And race 4 has lost the avirulence gene AVR-XV3. And this was surprising because the resistance targeting AVR-XV3 had not been commercially deployed. So we sequenced genomes of the tomato race 4 populations in 2006 and 2012 and found we don't have one single bacterial spot population, but in fact multiple different lineages of Xanthomonas perforans. And these lineages, at least in part, are differentiated by recombination events with uvesicotoria. So we have a diverse recombinant population. And then we looked at the mutations in AVR-XV3, and we found that there had been multiple mutations leading to the loss of this effector. And these mutations are correlated with lineage. So each lineage had lost this effector. So we didn't have one race shift. We had at least 4 race shifts. And I should say that I'm giving a quick overview of these results, but they'll all be presented in posters this afternoon, so I encourage you to talk to the students and postdocs who are actually doing this work. All right, so we didn't just see a race change, we also see changes in other traits. One of them is bacteriocin production. Some lineages have maintained bacteriocin production, and others have lost it or have reduced production. So this is another change that is also correlated with the demographic history of this pathogen. A change that is not correlated with the demographic history is the gain of another effector gene, AVR-BST, And we believe this isn't correlated with the lineages because this came in on a plasmid. And Peter Abrahamian, working with Gary Valade, has shown that this gene provides a fitness benefit in the field. So he knocked out the gene, put mutants and the wild-type strains in the field, and found that he recovered, over the course of the season, he recovered more of the wild-type strains than the mutants. I'm not sure why the mutant strain designation is cut off, but the blue and the green are the wild-type here, in the red and the purple are the mutants. So this is more than a race shift. This is many changes in the population. Does this represent, is this just Florida, or does this represent a global shift in bacterial spot populations? And this is something we're still trying to answer by gathering strains from production areas all over the world. But we do have some data from Nigeria. Our student, Mustafa Jibrin, sampled strains in 2014 and 2015. They looked weird. We sequenced genomes of representative strains, and sure enough, they're the diverged strains you can see the arrows pointing to in that phylogenetic tree. Again, recombinants, and I'm not going to show you that data, but you can see it on his poster. And one of these strains, we call NI1, has intermediate gene content between Xanthomonas perforans and Xanthomonas vesicatoria. It has had so much recombination that the genome is between those two species. The recombination has also affected secretion systems and type 3 effector genes. So these changes can have impacts on our resistance breeding strategies. So to summarize, we can begin to understand the changing landscape of plant pathogens by identifying what was responsible for the change in the pathogen population and why. And the perforans example shows that you can spend a lot of time just trying to categorize all those changes, and then we're still working on the why. You can determine potential pathways of movement and understand drivers of evolutionary change, particularly using population genetic approaches. Looking forward, we need local and global scales. So local surveillance would be really useful for our high-risk pathogens. And the medical community has shown the value of in-season, within epidemic surveillance. And with the lowering costs of sequencing, this is going to be feasible for us as well in the near future. But we need to combine this with global data through international cooperative efforts. So we also see the big picture, the geographic mosaic of the distribution of pathogens and their genetic variation. The real bottleneck at this point is in handling all this data. So thank you, coders and developers in the audience. We really need you going forward to make sense of all this data we're collecting. And with that, I'd like to thank all the people who contributed to this work, our funding sources, and my colleagues at UF.
Our final presenter this morning is Lav Kott, Assistant Professor in the Department of Biological Systems Engineering at the Irrigated Agriculture Research and Extension Center at Washington State University. Using case studies in specialty crops, his talk will discuss feasibility of the technology in field-level disease detection, as well as challenges that need further research before its commercial use. Please help me welcome Lav Kott.
Hi, folks. So before I begin, I just wanted to thank APS and Dr. Tim Murray, especially for inviting me here. It's an honor to speak to APS community, right? So just before I start, I want to say that I'm an ag engineer. So retrospectively, if I speak something wrong about pathogens and things like that, please bear with me, right? So we're going to talk about the state of art on sensing, right? Some of the technologies that are emerging in terms of plant disease detection, right? So again, most of my research is on, the bulk of it is ground and aerial sensing, and then develop data to decision support tools or technologies, right? So here we're going to focus specifically on the sensors. So what we're going to talk about is sensors, the optical sensors, the volatile sensors. As we are talking about optical sensors, we'll also talk about some of the sensing platforms, including drones, right? And then at the end, we'll summarize, just like we have, cell phones, so many apps on the cell phone, and we can control our homes, lights and things like that, like smart homes. Can we use some of that technology when it comes to doing something in agriculture, making it the concept of future of the farms feasible or not, right? So we'll talk about that. So most of the case studies I will be referring here are related to precision ag, few related to high throughput phenomics, right? So this slide here is to give introduction about crop sensing, right? And we have been doing this very effectively through scouting. You go to field, you see the, for example, vines there you are seeing some are healthy, some are disease, right? So you look at the vines and say, hey, this is probably a disease, or this is probably healthy. So if you have to implement the concept of sensing, how does it work, right? For example, you see there, we have this electromagnetic radiation from sun, right? As it is reflected, what we are sensing in this case is the reflectance that is coming out from the plants, right? So if and here, this graph here shows it, from 400 to 720 nanometers, somewhere there, if the plant is healthy, it's going to absorb most of the light, so reflectance is going to be less. That's not the same for the infected or disease or stress plant. It's going to reflect a little bit more. But from 720 onwards, there is a flip, right? The disease or stress plant in non-visible domain, it absorbs more light, so the reflectance is going to go down, and then opposite for the healthy plants. So that's the basic sensing principle if you have to use reflectance sensors and things like that. So if we have some pathogens in the plant and we have to detect this, there are different ways you can do this. You can use, for example, optical sensors. You can look at the reflectance change in the plant. We can look at the fluorescence change in the plant. If the plant is emitting volatiles, you can change in either change in the volatile concentration or different compounds that are emitted by the disease or healthy plant and things like that, right? So if you do that, then you have a sensing method detects the disease, and then you can do something to manage the disease. What we have been doing, I'm sorry, I supposed to go back. Before introducing sensors is we do use manual scouts, and very good example is Florida citrus. We have greening, and for several years we have been scouting the fields for citrus greening, right? Some of the other methods that we can use if we have to quantify the disease is PCR or ELSA, right? And we know that these methods, scouting is time-consuming. Also for some of the other methods, it takes a lot of time and energy to get the quantifiable data, right? So alternative is then used to use some non-invasive, non-contact rapid methods, and that's where our talk will be focusing on, using some of the optical sensors or the volatile sensors to detect the plant disease, right? And this is, again, an emerging field. There are very few options out there in terms of what sensors you can use and cannot use. So we'll try to talk about that, right? So when it comes to optical sensing, we have to start with what is called visible near infrared spectroscopy. And what you are seeing on the top here is the spectroscopic unit that looks at the spectral change in the object or the plant leaf for example, from 350 to 2,500 nanometers, right? Of course, it's not a cheap equipment. It's about $70,000 to have that equipment. The idea is, you use that equipment, look at the spectral change of healthy and diseased leaves and plants, and then you can develop something like this. This is a sensor that was developed in my prior lab that I worked in University of Florida to detect the citrus greening, right? So, looking from 350 to 2,500 nanometer domain, you look at the spectral change and find some bands, for example, 8 here, and then use that equipment or the sensor unit handheld to detect the disease, right? So that's a spectral response, and you get time series at each wavelength. You get spectral reflectance change, right? There's also hyperspectral field where we have instead of having the spectral response, you get at each wavelength you get the image that has spectral image, and what hyperspectral imaging gives you is a hypercube, very rich in information in terms of spectral resolution and everything, right? So you can use either spectral radiometer, or also you can use hyperspectral sensor, and the same concept. You can identify the spectral bands and then develop something that is low cost that can go out in the field to do the disease detection, right? Some other sensing tools, moving from vision IR to mid-infrared. You have spectrometer that can work in mid-infrared domain. We have thermal imaging that is very, from Department of Defense, it's taking over now. The sensors are going down from $40,000 to $6,000 and lower. This technology you can also use to do disease detection, right? Also, some of the active sensors, sensing tools that you can use, fluorescence imaging, LIBS, laser-induced background spectroscopy, or CT imaging. These are all active sensing techniques where you have this leaf or plant object. You have, in case of fluorescence imaging, you have leaf there, and you use the fluorescence to excite the plant molecules there, and then see the fluorescence change and things like that, right? And so you can use either of these based on your application and things like that, right? So as we're talking about the optical sensors, it's logical to talk about the platforms. There can be different types. For example, if you are using vision IR, you can use fiber optic probe to clamp to the leaf and get the spectral response without detaching the leaves from the plant. So that's handheld platform. You can also use agricultural vehicles like this, and we use a lot of this in our research. We have different sensors, including LiDAR and all these things. We mount on the gator and get the mapping done at a high throughput rate, right? For practical sense, of course, you cannot have this retractable mask and things like that, but you can have the vehicles that are used by the Scout. You can also use tractors and mount these sensors and do high throughput sensing there. And again, for the last few years, we have been talking about the drones, the small, especially less than 55 pounds, how you can use that for doing the remote sensing, right? And also, there's another field of that is not for detection, but rather management. Like there is emerging field about using the mid-size unmanned aerial systems, which can be used once you do sensing, identify the infected area or the block within a block. You can use this technology to do surgical spray applications to manage the disease and things like that, right? So as we are talking about drones, let me a little bit talk about why we're talking about drones, right? And the bottom line here is the resolution, right? It's not new. That remote sensing, we are doing for several years. We are getting the data from satellites at 30 meter per pixel resolution, but we always complain that there is not enough resolution to look at the plant leaf and things like that, right? The good thing about using drones is, you can fly this just above the canopy and get, the resolution that you need to look at certain disease and things like that, right? And again, the frequency of data acquisition, satellites give you data every 14 days. Drone, you can fly anytime you want and then get the data that you need, right? So a little bit about regulations. I'm just going to go quickly. It's just like buying a car now since last October, right? Last September. You buy a car, you buy a drone, you get a license for driving your car, you get a license, what is called US pilot license to fly a drone. and you are good to go as long as you understand the regulations. Previously, it used to be a little bit tedious, but now it's a lot simpler to get a drone and fly it, right? And in fact, you can go to Best Buy and some of the other stores and get the consumer-grade drone. In terms of flying, you can still need to be in, you still need to be in the line of sight. Vertically, you need to be about 400 feet or lower. And again, you need to be 16 or older for flying the drones, right? If you are a government entity, for example, USDA, or any other public entity, you can follow the regulations that we discussed earlier, Part 107, or you can just get the blanket COA, Certificate of Authorization, and you are good to go throughout US in Class G airspace, right? Also, in this case, what you see here is you can self-attest your pilots. That means you don't need to get the pilot certification there, right? And these all are for the weight that is less than 55 pounds. If your drone is going to go higher than this payload with your optics and everything, then you need to get what is called 3.3 exemption, which is a little bit tedious, but you can still get it, right? So that's about the regulations. This slide here quickly is, because we talk a lot about drones and we think that drone can do so many things, the bottom line is drone is only one of the solutions, not all the things, right? We have been using satellites. There's also the field of what is called low orbiting satellites. Last 2 years, there's a company in California and Europe. They are launching. Our satellites ideally are in a higher orbit. They are launching these low orbiting satellites, and they're giving the data every other day at the resolution of 1 to 3 meters per pixel, right? So you are using the satellite data. You can use low orbiting satellite data and then fly the drone only if you have to in the regions that you see some problems, right? And that's how drone has to go and integrate in terms of using this for practical purposes, right? Again, last few years, when we started talking about drones, we always started, like there were so many companies that were developing drones. And then soon we realized that, hey, we have drones, but what kind of optics we can put there to sense something, right? And that's where in the last few years, we are developing range of sensors. Some companies are developing this that can be integrated on these small and manual systems and all these things. But then again, if farmer has to use this, it's difficult to fly this every day and get all this information. So ideally, ideal integration is then crop service providers then use this technology as an additional tool to identify the problems and talk to grower or farmer that, hey, this is additional information that we are getting from this sensing, and use the drone as just a tractor. You can have different sensors that they're as per the need and things like that, right? And that's how it has to be meaningfully integrated into the agricultural applications. Going back to the sensors, quickly we are going to go through some optical sensing techniques. This is one of the Vision IR sensor I talked about, right? Again, it's looking from 350 to 2,500 nanometers. And I'm going to go back to the example of citrus screening from our prior lab. So as I said, you're going to get change in reflectance. And you can see here the healthy leaf has less reflectance here, higher here, and things like that. And you can compare the reflectance data for the canker and greening. And then based on this, going from 215 bands, you can develop a sensor that goes on the drone that has 8 bands or 6 bands rather here, and do the scouting for greening detection, right? And so there are several publications from the lab that I worked there in the University of Florida. How we are using this technology in Washington is, Dr. Naidu, he might be here somewhere. He has done some work in 2006 where he looked at this GLD, leaf roll disease. So what he did is he detached the leaves from the vines a little bit late in the season and get the spectral response, right? So since I went there in 2013, what we are doing some preliminary basis is, without detaching the leaves, we are just taking this Vision IR, getting the spectral response, and seeing whether we are getting the same response or not when we detach leaves or not detach leaves, right? And so doing all this, you can see we can clearly separate the infected and non-infected leaf. And these are some of the bands that possibly can be used to develop a sensor, right? But that's not the end of story. There's a lot of research that need to happen before we develop a sensor, right? So because we have hyperspectral sensor, we are going one step ahead and developing, or looking at the spectral response using hyperspectral imaging, right? And so again, hyperspectral imaging here, what you see here is for each wavelength, instead of getting a spectra, you are getting an image of that leaf, right? So there's a lot of data. We call it hypercube, right? So the challenge with hyperspectral sensing is it's information rich. At the same time, you need to have very good computing resources and very good, expertise or very technical expertise in mining that data to understand the plant response that is gathered through the hypercube, right? Again, in terms of sensing, there are different types. For example, for several years, several companies have developed the push broom technology where, you have the field that stays stationary, but then you have a sensor moving, and then it's scanning each line, and then it's gathered as an image, and that line scan then is your hypercube, right? For last couple of years, there's a lot of push about developing what is called, like, your cell phone gets a snapshot, developing the hyperspectral sensing that does the snapshot imaging, right? But frankly speaking, we are not there yet in terms of using that technology for, disease detection because of several issues that are there with snapshot imaging, right? Still, the push broom is one of the better ones. Again, the cost of hyperspectral is, depending on what sensor you buy, the one we have is about $100,000. And that's the sensor here you see here. So again, what we are looking at is, spectral response from the leaves, and then unlike last, study that I talked about, we are going a little bit early in the season to see can we detect GLD at early stage, right? And so this all data processing, all these things, but we are looking at these bands. Are they same as what was discovered with prior studies and things like that? So again, to have meaningful sensor development, what need to happen is you take these bands for GLD, then you have other disease in the field, look at the spectral response there and all these things, and then still see the validity of those bands, and then develop a sensor that could go on the agricultural vehicle or on the drone or something else to do the high throughput mapping there, right? So this is about a little bit about mid-infrared. Again, what we are looking with mid-infrared is a chemical composition. For example, in here, again, going back to citrus greening, you are looking at the relating this response in micrometer range, absorbance of the mid-infrared to relate that to the starch accumulation in the leaves, and that can be related to the citrus greening, right? And that has been, well-researched technology there. Also, as I said, the mid-infrared, the infrared thermal imaging, it's one of the emerging technique that, you can use. It gives you a pixelated temperature. For example, in this image, what you see here, each pixel, you are getting a temperature here, right? So you can use this, and this is the sensor that we put on the drone and get this map here, right? You can see the soil temperature and the plant canopy temperature. You can use that information with some other sensing tools to map the disease infection in your field and things like that, right? So this is quickly another example. This is not disease detection, but rather we are looking at the knowing that climate change will be there. The USDA researchers, they are looking at different cultivars that will be sustained. Which one of these will be sustained if there is less water and hot climate temperature in Washington. And so what we are doing is these central 2 rows. We have 8 cultivars again replicated, but then they are getting water at 50% of the required rate. And here you can see the cooler canopies that are getting the water at 100% of the required. And so, and we are again using the drone-based NDVI and thermal imaging to look at the progression, which varieties are doing better in terms of crop vigor and water stress, and relating that to the yield and all these things, right? This is another example where we are looking at using again drone imagery where we are getting NDVI and thermal imagery where we are looking at the blight infection, or the blight resistance rather, in chickpeas, right? And this is just not to just do detection, rather it's to help some of you guys, if you are going and rating your plots in the field with just manual ratings, you can use drone imagery to reduce your time and things like that in the field. And this is rather a phenomic application here, right? The fluorescence imaging, again, it's active type. You have the leaf, you have excitation wavelengths, and then get the fluorescence, some of these ratios that you can use to get the disease detection done, right? A word of caution here for fluorescence imaging and any other imaging that you want to use, right? For example, here, what these researchers found here is, they look at the hamlin citrus leaves in Florida and the same variety in Brazil, and they have different or low fluorescence in the leaves from the trees that are in Florida, right? So for any sensing, if you have to use, depending on the location of your crop and the region, the reflectance and some other spectral responses are going to change. So you have to have right calibration model for your sensor to detect the disease. There's no one solution or one shoe that fits all, right? So that's how we have to go if you have to use non-contact sensing for doing disease detection. And so this is, in summary, I'm going to skip this for now. So quickly, we'll talk about plant volatile sensing, right? And we know that the plants, when they communicate with each other, if there is a disease, infection, things like that, they do emit some volatile organic compounds. So just like the optical sensors that look at the plant reflectance, emission, things like that, we can also look at the volatiles that are released by the plants, right? For example, if their plants are under stress with some disease, they release some compounds like this, volatile organic compounds. If they are due to the water stress and things like that, they release some different organic compounds. So in terms of sensing then, for several years, we have been doing this spectroscopy, or spectrometry rather, right? GCMS and some other variants of spectrometries where our spectrometries rather, where you get this volatile absorb onto SPME or things like that, and then run through the GC and then the column. Then you get the mass separation and get the signatures for certain volatile organic compounds, right? For today's talk, I'm going to focus heavily on introducing this differential ion mobile spectrometer. This is the technology that is used by DoD when they have to detect mines and things like that in the war field. If you check in your bags and when you're or when you're crossing the different ports, airports, you see if your bag is swabbed, and then they use this spectrometry to look at the explosive compounds, and things like that. So same technology can be used for plant volatile detection, right? Also, there are several types of electronic nose-based sensing arrays that have been developed and things like that. The problem with electronic nodes, again, is a lot of research in terms of food safety and all, but the issue is humidity and all other aspects that makes it less viable a candidate for using this in agricultural plant environment, right? But we have found that the ion mobile spectrometer that we have, and let's jump into that one, is one of the best candidates that so far that is giving good data to us. The way it works is this is the ion mobile spectrometer. We call it FAIMS. What we are doing, and this is again post-harvest study rather, we have these tubers infected with soft rot, and then we are just using the carrier gas, pushing the volatiles from here to the column, and then in here what we have is ionization. So we ionize these molecules, and then the molecules will be drifting through this tube, and we are looking at for different voltages, you are looking at this ion current variation, right? And so you can see the ion current variation here, and then you can effectively train this sensing unit for detecting certain diseases, right? So we did this to detect the potato soft rot at post-harvest level. We have what you can see here is the on day 0 when we inoculate tubers, healthy and infected ones have the same response. But on day 1, again this is at room temperature by the way, you can see some change in the DF matrix response there, and that's telling us that there is something that there in terms of volatile change. And then we run some standards and identify what are those volatiles that can be related to the soft rot, right? So the same note, again, you can use this for detecting plant volatiles. This is another study done by another group in Washington State. What we are looking at is 3 different varieties of the chickpeas, or not chickpeas, rather peas. 2 are resistant to fusarium, and 1 is susceptible, right? And we are looking at the volatile on every other day, and then you can see, at 1 week you are getting some response, but in 3 weeks you can clearly separate those volatile responses there, right? So the problem with one of these thing is not the sensing, rather how can you collect those volatiles, right? And so there need to be some little bit more advancements in terms of sampling those volatiles and transferring those to the sensor, right? That's one of the challenge that we have as we are doing this. So this is last slide, by the way. This talks about Internet of Things, smart agriculture, right? So we can develop so many sensors and so many things, and still we need to go out in the field and collect the data, right? So through this concept, you probably don't have to, right? So what this is about is just like cell phones having different apps to control several things in your home. You can have independent servers, and these are low-cost servers. For example, what we have developed in our research is this is a Raspberry Pi computer that costs you about $80, and we have this thermal infrared and RGB imaging. Total cost of this one is about $400, right? So each server in the field. Can you please play the video? So you can have this in the field on a tower, and you can monitor your crop. This is thermal image there, right? And this is just an example. You can have any other sensor that's out there talking to these servers, and you're collecting data, and then transferring that data to remote sites, and you look at this data and say, hey, there is something wrong that is going on in my field. I need to take some actions and things like that, right? Again, we know, and the wireless sensor networks are not new. The issue that we have in agriculture is the connectivity, right? There is no 3G, 4G, or 5G connectivity in the remote locations on the big farms that we have in Washington and some other states. So there is a big push independently from the Microsoft and some other companies to use what is called the TV white space. TV white space is the bands that previously, before the cable and some other technology came in, we used to have this big antenna on the tall antenna on our homes and get the channels, right? So those bands, because we moved to digital arena, we have not been using those bands. So Microsoft is developing some technology, hardware, that we can harness that white space to transfer the data remotely and long distance. And so that's the future that we're going to see a lot of this hardware coming into agriculture to do the machine-to-machine communication, so that we can use that for not only sensing but do something in return to manage the crop loss, right? So with that, I would like to stop here. And I'm not sure how much time we have for questions, but thank you again for being patient with me.
So I had a question for the last speaker. So if you can identify diagnostic wavelengths that target your disease of interest, being more biology than engineering, how do you take it the next step and get a sensor built?
Okay, so the question is once we have the bands, how can you take it to the next level to develop a sensor? Is
yes.
that right? Okay. So again, it's not easy, right? As it sound, or I made it sound, right? So we have the bands that are identified for one disease. Then you go to the field and identify, look at the validity of those bands and at field level, right? Because reflectance in the lab and in the field, for example, is going to have different effects and things like that. So once you find the validity or robustness of those bands, then again, for a given crop, for example, we have grape vine, you introduce some other diseases there and see the spectral change that's happening because of those combinations. And then validate those bands, keep validating it, and then you will come up with 6 or 8 or say 4 bands. Then you talk to the sensor or some companies. for example, that was done in Florida, you give these specific bands to a company and say, hey, can you develop the optical sensor for those, right? The good news is, if you have the bands less than 1100 nanometers, the cost of optics is less. But if you have bands that are beyond 1100 nanometers, the cost is going to be a little bit high-end because they have to use different types of detectors, right? And then once you have that sensor that is customized, you can develop in your lab or you can get it custom developed. Then again, you need to go back to the field and see whether that sensor is doing what it's supposed to, as was identified by the ground truth or base equipments, hyperspectral imaging or Vision IR, right? And if that is the case, then you can keep using it for doing crop scouting. So it's a life cycle. It takes some time. But there are some sensors, for example, MicaSense and the FLIR. There are some other companies who develop these custom sensors. As long as you do good research identifying those bands, you can develop sensors.
Thanks. We have another question.
I want a question for Mr. Koth. Can we use the volatile sensing technology for seed health testing?
So the question is, can we use the volatile sensing for seed infection?
Seed health testing.
Seed health.
Yeah.
So as long as
you can
there is
differentiate
emission
between
of volatile organic compounds, you can use the sensing, right? So the way it works is you either see the same volatiles or volatile organic compounds or VOCs released at different concentrations with healthy and infected seeds, or maybe there is some different volatiles that are released by the infected ones compared to healthy ones. So the answer, short answer is yes, you can.
Yeah.
The challenge is trapping those volatiles, and probably with seed, it might be easy to trap the volatiles than in the plants as they are growing. Yeah.
Thank you.
So I have a question for Greg. Greg, it was a very good talk. So I just wondering, because you have tried both transgenic-based host-induced gene silencing as well as RNA-based external application of RNAs, which one you think is more efficient? And then the second question is, how did you quantify the double-strand RNA levels in the transgenic plants? Because I assume all the double-strand RNA should be processed into small RNAs already by the plant small RNA machinery.
Okay, so there's two questions there. One which is more efficient, and I would have to say it depends. That's my lawyer answer, but it's really going to depend on the context of what you do. So take the example of Colorado potato beetle. It's highly responsive to surface-applied double-stranded RNA. You can, in theory, engineer a plant that it might feed on like a potato and also generate that double-stranded RNA in place. Both, I think, are equally efficient. The cost and effort in generating and deregulating a transgenic plant is probably going to be different than a topical surface application, but you still have to figure out how to produce that much double-stranded RNA because you're going to need more. It's going to be less efficiently applied on a broad acre scale. So you've got a little balance there. And the other thing, too, is with a topical application, you have to look at the window. So the plant will, be basically shedding this through the action of UV, microbes, things like that, will all be assaulting the RNA that you apply. So you have to have it long enough that the larvae can encounter it in a meaningful way for a long enough period of time. So a surface application that might need to be applied very frequently might not be practical. So it's just going to depend. And we find that the real world, the results we see in field tests versus what we see in a growth chamber can be radically different. RNA tends to be a lot more stable in a controlled environment than in the field. And so you're going to have to manage that ability to deliver the RNA through a topical application. Not impossible, but it's probably going to be more challenging. And so we'll go from there. As far as the quantification, we have one of the publications that was actually on one of that slide talking about the detection in the environment, we actually had to refine some assays to look at double-stranded RNA detection in Planta for the long double-stranded RNAs because the resident methodologies weren't quite as good. That snapback, the ability to fold and make that double-stranded RNA actually stymied some of the easy applications for quantification. But we were able to get past that. And the thing is that in Planta, it's not all processed. It's relatively slow in a plant like corn. So there's a large pool of unprocessed double-stranded RNA, and it gets processed to small RNAs, but it's a slow enough process that rootworm perceives it and imports that. It won't touch the small RNAs, but it will perceive the large.
So you think the worm require the long double-strand RNA for efficient silencing?
Long double-strand RNA is needed for something like an insect. We've never seen small RNAs functioning in presentation scenarios, whether topical or in Planta. So I can go through that data, but we don't see small RNAs entering even from a plant into the insect.
Mm-hmm. And have you tried the nanotechnology, the clay-based protection of the external RNAs?
Yeah, so there's been some recent reports on binding RNAs to clays and then hopefully using those to promote longevity in the environment and have a distributed load against a viral target, for instance. And so that's very intriguing work. We have not internally tried to replicate that at this time, so I really can't say how it compares to the other things that we have tried. The only thing I think in that report was, again, all of those studies, I believe, were done in controlled environment, which again, we see quite different outcomes in terms of longevity versus in field scenarios.
One question for Greg. Your research showed an excellent example of managing like a disease caused by like more of an entomology disease. As a plant pathology, I wonder what are some of the opportunities as well as challenges in terms of using RNA silencing in managing plant pathogens such as bacterial pathogens, fungal pathogens, as well as viruses. Thank you.
Well, I think the track record so far, both what's actually in use and what has been done in laboratory scenarios, say that viruses in general are extremely tractable. So again, it's knowing your target, knowing the sequence, the diversity in your viral populations and so forth, will really sort of enable you. But that's doable in my opinion. Fungal pathogens, there's some reports in the literature, but I think you're going to see a case-by-case basis there as to what really is tractable. Necrotrophic fungi, I think, are going to be really hard to approach in a robust field scenario, because they're secreting a lot of hydrolytic enzymes, and at least from my insect examples, those hydrolytic enzymes are pretty much deal breakers in an easy application scenario. And then when you get to bacteria, I would say, we don't have really good evidence, I think, of those kinds of scenarios where people have approached bacterial targets. Doesn't mean it's impossible, but the systems are a little bit different. They don't have the canonical RNAi machinery. They do a little bit different type of regulation, so you would have to figure out how you could get into the bacteria, A, and then B, what would work in that scenario. And it was probably going to look different from what we can do in eukaryotic organisms.
Okay, I want to thank our speakers again for some great presentations.